I think microbes are the coolest. Microbes are these tiny little chemical factories that have evolved to live in pretty much every environment on Earth. They also fix nitrogen and phosphorus, which means that they can convert it from forms that plants can't use to forms that they can. Nitrogen is a major component of the air we breathe. It's essential for plants, and actually it's essential for us too. In order for both humans and plants to make those molecules that are essential for life, they have to have nitrogen in a form that they can use, and nitrogen in gas form is what's called inert. What we need are forms of this atom that we can actually use chemically. So modern agriculture is founded basically on this process called the Haber-Bosch process, which is amazing. It's this industrial process that allows us to make ammonia. It's responsible for feeding about half of the global population. The problem is it's responsible for about 2% of CO2 emissions globally. Not only that, but it's generally done in large factories, and so the entire continent of Africa does not have a plant that can make ammonia. So they have to import all of their fertilizer. Not only is this contributing to climate change, which we know impacts people in the global south more than in developed nations, it also is just more costly to be a farmer there. My lab generally focuses on energy equity, which is the concept that marginalized and disadvantaged groups have often been left out of technology, and so it's our job as researchers to develop accessible, affordable technologies for them. Microbes basically support all life on Earth. We're actually more microbe than human. We have more microbial cells in and on us than we do human cells. Without chemical fertilizers, plants grow because of interactions with microbes. The process of converting this inert gas into forms of nitrogen that plants can use is something microbes are really good at, and this whole process is called nitrogen fixation. And they also do things like secrete pesticides, so they can actually protect plants from pathogens. So they do everything that we try to do with chemicals, but they do it better, more efficiently, and locally. In their natural environments, microbes live in communities where they support each other. They're basically like a family in a house. But when we take them out of this environment and we want to scale them for production, they're exposed to all these stressors and they don't like that. So it can be too hot. It can also be too cold. There can be too high pressure, too low pressure, like a vacuum. They also don't like being in liquids that aren't water, and all of these things kill the microbes. Microbes need to be alive in order to work in soil. So in order to replace agrochemicals, we have to make sure they're alive at the time of delivery. Most cellular processes are based on these reactions called Redox reactions, which are basically just electrons moving between molecules. This is the basis of how we function and how plants function for both respiration and photosynthesis. If an electron is removed, it's called oxidation. If it's added, it's called reduction. And we can kind of squish these two words together into redox reactions. One of the main ways that all of these different stressors harm microbes is through oxidative or reductive stressors. And so when we think about protecting cells, a lot of the protection we need is protecting them against these reactions. The challenge of keeping microbes alive is what my lab sought to address. And the way we thought about this was Protecting these microbes from both physical and chemical stressors required basically a pretty versatile suit of armor. We didn't want to add chemicals that might be actually more detrimental. And so the materials we settled on are antioxidant compounds. One of the main coatings we use to protect these microbes is a combination of iron in its 3+ form and tannic acid, which is a component of tannins found in coffee and tea. So when these coatings originally form on the microbes, you get these loose interactions between OH groups on the tannic acid. Then we add a base, and that removes the proton. It removes the hydrogen from the OH group, and it rigidifies the coating, kind of like an Iron Man suit forming on Tony Stark but not completely closing. That's the blue color. And then once it rigidifies and completely protects him, it's the equivalent of us adding the base and seeing that change from blue to purple. Now that we have these microbes and we know we can store them and keep them alive under these conditions that are not ideal for them, we wanted to see how they actually affect plant growth. One of the ways to test that is to actually add these microbes to seeds that you're going to plant. We consistently see across all of these different crops about 150% higher germination with the proper microbe that's been coated and stressed compared to the fresh microbes. And so we were really excited by these results because it would allow a much broader swath of the farming population to actually use these materials. In addition to kind of using microbes directly for helping plants, we've also protected microbes that can do things like take CO2 from the atmosphere and mix it with calcium and make limestone. And it allows you to change the pH of soil without carbon-intensive trucking. Improving vaccines so that we have more access to vaccines because you don't need to store them in a freezer. You can store them at room temperature. Microbes can do everything from produce rust to remove it, so we can use microbes to protect building materials. Science is more than textbooks, and you can actually make real-world impact by doing research. Our goal is to push the frontiers of what we know and what we can do by using what biology has given us. I see chemistry as the foundation of every other form of science. If you understand how molecules are built, that explains biology, it explains a lot of physics. It basically ties everything we know about the world together.